Have you ever wondered how we could get new medicines to people faster, cheaper, and with way less risk? What if the secret to doing that wasn't just in a test tube, but was actually hiding inside a computer? Well, today we're going to pull back the curtain on the absolutely incredible world of digital drug design.
I mean, really think about it. That simple pill in your medicine cabinet has been on an absolutely epic journey. So what does that journey actually look like? Well, you might picture a straight line, but the reality? It's much more like a giant, sprawling, and super complicated maze.
And this maze is, well, it's brutal. We're talking about a process that can take decades. It can cost billions of dollars. And the really tough part? The vast majority of promising new drugs, they fail. They never even get a chance to help anyone. There's got to be a better way, right?
And this is where things get really interesting. What if, instead of just stumbling around in that maze, we could build a digital map, a map that simulates the entire path on a computer, guiding us around the dead ends and showing us the fastest way through? That's the game changer we're talking about.
Okay, so you want to draw this amazing digital map. Where in the world do you start? Well, the very first step is to figure out what the most important landmarks are. And for that, we have a tool that helps us find what you could call the critical levers.
Now, the name, parameter sensitivity analysis, I know, it sounds super technical, but the idea behind it is actually pretty simple. Imagine a giant machine with 100 different dials and knobs. All this technique does is methodically turn one dial at a time to see which one has the biggest impact. It's all about finding the stuff that really matters.
And the process itself is really systematic and smart. First, you set a baseline, where everything is just at its normal default setting. Then, and this is the key, you change just one little thing. Maybe it's the drug's particle size or how quickly it dissolves. And finally, you just watch and measure the impact. Simple, right? But it's an incredibly powerful way to understand a complex system.
So why go through all this trouble? Well, the payoff is huge. When you know which dials are the most important, you can design a better drug right from the very start. You can figure out what's holding it back, and you save a ton of time and money by not wasting resources on the things that don't make a difference.
All right, so we've figured out which dials are the most important ones to turn. The next logical question is, how do we test our ideas? Well, this is where it gets kind of futuristic. We get to run trials on thousands of virtual patients, simulating real-world results with zero real-world risk.
Yep, you heard that right. Before a single human volunteer is ever involved, researchers can run what are called in silico trials. It's just a fancy term for running trials on a computer. They create thousands of digital profiles that represent a huge, diverse population and see how the drug behaves.
And just look at the difference here. It's staggering. Real-world trials take years. Virtual trials can be done in days or even hours. The cost plummets from billions down to next to nothing. And you can test on a simulated population that's way bigger and more diverse than you could ever realistically recruit. This is a massive leap forward. And don't think for a second these are simple simulations. We're talking about modeling incredibly complex systems inside the human body. Things like the changing pH levels in your gut, how fast things move through your organs, how a drug binds to proteins in your blood. It's this insane level of detail that makes these virtual trials so predictive and so powerful.
Okay, quick recap. We've found the key factors, and we've tested them on an army of virtual people. But there's still kind of one piece of the puzzle missing, right? How do you reliably connect what you see happening in a lab beaker to what's actually going to happen inside a living, breathing person? For that, you need a translator. And that translator has a name: in vitro in vivo correlation, or for short, IVIVC. Let's break that down. In vitro is just science speak for in the lab. In vivo means in a living organism. So IVIVC is basically a mathematical bridge that connects lab results to real body results.
Think of this bridge like a two-way street. Going one way, we have what's called convolution. This is all about prediction. You take your data from the lab, you plug it into the model, and it spits out a forecast for what will happen in the human body. It's basically letting us peek into the future, all digitally. Now for the other side of the street, deconvolution. Here, we work backwards. We take the data we got from actual human studies, and we use the model to figure out what the perfect lab test should have looked like to predict that outcome. This creates this amazing feedback loop that makes our lab tests better and better over time.
So, we've found the key variables. We've run virtual trials. We've built this incredible bridge between the lab and the human body. What's the end game here? What is the ultimate prize for all of this amazing digital work? Well, it's the ultimate digital shortcut. Get this, the goal is to be able to skip certain human trials, safely of course. When your computer models and your lab data are so good, so predictive, you can actually demonstrate that a drug is safe and effective without needing to run another massive, expensive human trial. This isn't some rogue idea either. It's a formal, regulated process called a biowaiver. It's when a regulatory agency like the FDA officially agrees that your computer modeling and lab data are strong enough to substitute for a specific human study. It's not about cutting corners, it's about being way smarter with the science.
And the benefits here bring us right back to where we started. Biowaivers/development time. They save incredible amounts of money. But maybe most importantly, they're a huge ethical step forward, because they reduce the number of human volunteers we need to expose to experimental drugs.
So there you have it. That's our digital toolbox for designing drugs in the modern age. You start with parameter sensitivity analysis to find what's critical. You use virtual trials to test your ideas safely on a massive scale. You use IVIVC to translate between the lab and the body. And all of that can lead to a biowaiver, the ultimate shortcut.
And all of this leads to one final huge question. As these tools get even more powerful, as our digital maps get more and more accurate, are we getting closer to a future where a new life-saving medicine could be designed, tested, and approved almost entirely inside a computer? Now that is something to think about.